Hello, everyone. So welcome to our seminar today. So we're so happy to bring you guys today, Dr. Saag. And just as a reminder, or just to talk a little bit about, what has been happening in the Sigma Xi world. Thank you so much for being here and for your membership. So we have some news from National. So if you would like to join Sigma Xi, you can join or renew your membership. And if you need any help, if you are from the UAB chapter and we want us to help you with that, just send us an email. Remember, we have the annual meeting coming up and it's going to be virtual this time. And the theme is Hacking the Brain, the intersection of art and neuroscience. They will have even a STEM art and film festival. So just go ahead and check that out. And if you are an undergraduate student or graduate student, there is an opportunity to apply for grants and aid, but the deadline is October 1st. On our side here, we have a new vice president, that is Dr. Lauren Rest. And our next seminar she's an assistant professor in the Department of Physics. She works mostly in physics education and has a background in computational physics. Did I say that right?
Yep, that's right.
So our next seminar, it's going to be on October 21st, and we'll have Dr. Jean Siegel. And then we'll have another in November that will send the announcement, and it's going to be Dr. Lauren Rest. So stay connected with us. We send announcements about our seminars to the members, the chapter members, and you can also follow us on Facebook at Sigma Xi UAB and also send us an email. But today, right, we are here to hear Dr. Saag. And I'm just going to give you some instructions on how this is going to happen. Usually we have these meetings in person, but today we're doing this virtually. Thank you so much for the Sigma Xi headquarters to helping us with that. So we'll have a few, some time for questions at the end of the presentation. So, but please submit your questions in the Q&A box. And if you want to receive emails from us about our next seminars or anything, please email us at sigmaxiuab@gmail.com. We usually have a signing sheet that we keep, but today, if you can email that, that will be great. And once we will try to finish this a little bit before 1 o'clock, but we want to have like 2 minutes because we have a very big announcement that we want to make, and we want you guys to stick around for that. So you are here not to hear from me, but from Dr. Saag. Dr. Saag is the Associate Dean for Global Health. He's the director of our UAB Center for AIDS Research. He's a professor of medicine. He has published so many publications, some of them very important in the area of, virology, HIV. He also is a book writer. He also wrote a memoir. So, I mean, if I don't want to take any time away from Dr. Saag, just, highlighting his CV, but he is great. And we are so happy that he is here with us. So you guys are in for a treat today. Great. So without any more delay, I'll stop sharing so that you can share your slides.
Okay, great. Well, thank you so much, and it's great to be with everybody. And I'm going to share screen here and get this started. And let's get the slideshow. There we go. So this is an overview of COVID, everything you wanted to know about COVID, but we're afraid to ask because there often is no answer. And I think that's about as good as I can summarize what this epidemic has been like for all of us. And it's amazing how much we know in a very short period of time. The epidemiology is what I'm going to review first, and transmission dynamics, treatment and prevention, and then something called long COVID, which is a new frontier. let's go through the epidemiology. Basically, it's bad. This is a little bit outdated, but the United States went up, came down a little bit, went back up, came down a little bit. Now it's going back up again. This is not as updated as I'd like, but the bottom line is there's roughly between around 50,000 new cases per day and somewhere around 1,000 cases or less of people who die daily in the United States. And one other way to look at this is in terms of Alabama. We have been up as high as almost 2,000 cases a day. About 4 weeks ago, we were at 1,600. We're probably down to about 1,000 cases a day. But what's more important is how many cases do we have per 100,000, and that's about 20 or so right now. And the case positivity rate, in other words, of every test we run, how many are positive? And that answer is about 16%. And where we want that is somewhere around 5% or less. New York City, for example, is 0.9% positivity. That's a relatively safe environment. That's what we should aspire to do. This is a little bit dated, but I think you get the point that if you compare the US curve, which kind of looks like the overall curve above, and this again is outdated, but if you just want to sort of look at what the US kind of looks like as reflected in an indirect way here, most other countries have gotten it under control. We have not. We're probably the only industrialized country in the world that has not gotten this under control. Sweden also, more through a deliberate approach, didn't, but everybody else did. The problem is that in a lot of these countries today, in particular the United Kingdom is starting to go back up again. It's not reflected in this, but it is. Germany had a little bit of an increase. France is now experiencing a little bit of that. China, remarkably, if you believe the numbers, came down, and their total caseload is 90,000. And it's been it's been negligible the last month or two. And you could question that, but actually I think it's probably correct. Israel is an interesting story where they had a case burst. It came down nicely. They developed technology that's pretty spectacular to do case contact tracing, and then they relaxed, and now their case rate is the highest in the world right now per 100,000. Just boom. So it just goes to show that when you get things under control, you can't get overconfident. You've got to stay with it, and we're in this, the virus doesn't care. The virus just wants to infect people, period. That's what its mission is. And our mission is to keep that at bay. And the sad news from my perspective is that we know, and I'll go over this in a minute, we know exactly how we can mitigate this. We can do exactly what a lot of these other countries, and for that matter, New York has done, or Vermont has done, or Montana has done. They've gotten it under control. But unfortunately, most states in the southeastern US and now in the Midwest are not doing as well. And I think it's because we aren't applying our knowledge in the most optimal way. So how is it transmitted? It's a virus, but let me tell you a story about me. So this is a story where I had, at the beginning of March, gone to a meeting in Boston, and it was supposed to be a huge meeting, and it got canceled to become a virtual meeting about the moment my plane touched down in Boston. So I had a decision to make. I actually worked with the organization that led the meeting, so I volunteered to stick around and help the meeting out. And so we went through, converted this from a live meeting to a virtual meeting in literally 36 hours, quite a feat, and pulled it off, and it went well. And my son, who's a physician who lives in New York and Manhattan, wanted to come back home for a couple weeks and wanted to take his dog. He's going to drive. And I said, well, I'll take the train from Boston and drive back with you, which I did. And at that time, if you recall, the thought about transmission was that it was mostly through contact, touching a surface, touching your face, that type of thing. But airborne was really not suspected. So we were very assiduous about keeping our car clean and wiping things down. but we're sitting in this car for 20 hours together, not wearing masks, and it turns out that he, unbeknownst to either of us, was infected. And so by the time we pulled into our driveway in Birmingham on Friday night, he said, I don't feel real well, and he took his temperature as 102. We looked at each other and said, uh-oh. And by the next day, I started getting symptoms, and on that Monday, we both tested positive. His course was about 6 days worth of kind of feeling bad, but not too bad. My course was different. I had about 6 days of feeling bad, but not too bad. And then on day 0, I am quarantining what I called Rikers Island in my house, and just sort of hanging out there doing emails or whatever I normally do. And this was another look, and I'll point out, you'll notice a pulse oximeter here, coffee, of course, my computer, paper plates, gloves, but not too much in the way of masks, because we didn't know in the second week of March that was important. Later went on to develop what I call a Groundhog's Day scenario, where by day 7, I was fine in the morning. 5, 6 o'clock at night, I started to feel bad and had what I would term a cytokine storm, where I get fatigued, headache, fever, fuzzy thinking, I call it, just totally very ill. And my pulse ox would drop to about 90, and I would be on the precipice of where I would say, do I need to go to the hospital now? And then the next morning I'd be better and say, oh, okay, well, I survived that. And then like Sisyphus, by the next night, the fever, everything returned just as bad. And the horror of it was that I wasn't sure if or when it would ever go away, but even more so, would there come a time when I would get so sick that I'd have to go to the hospital? And I've thought about this a lot since then, because, you know, it's interesting. I never really thought about dying. That wasn't like a fear that I had. If I died, I guess I died, and that would be unfortunate for obvious reasons. But I was more afraid or more concerned about going into the hospital, because I would be isolated. I might have to go on a ventilator, which I didn't want to do. And that was really the driving fear that I had. Fortunately, it never came to that. I went through 8 days of what I call Groundhog's Day, morning okay, night's horrible, and then it just went away and didn't come back. And now I'm working in the COVID clinic, seeing patients, and I also donated plasma because my antibody titers were the highest they had seen. So I donated plasma and cells through leukapheresis back in May. And so that was for science, that was for research purposes. But I learned a lot from having been an AIDS doctor in the 80s about new epidemics and what works. And the number one thing I learned was that it was very important to participate in science. That's how we're going to make progress. The second thing I learned was to go public with my story. And so I've been really trying hard to get the word out about the badness of this epidemic and trying to fight through some of the political maelstorms that we have, where we're not really getting clear messaging, and that's unfortunate. And that's, I think, one of the reasons, in fact, probably in my opinion, the reason that our numbers are still up here and high and the rest of the world is low, because we've allowed, or we've gotten into a situation where politics is interfering with public policy. And the final thing I learned from patients was how hard it was to be sick when there's no known therapy. And the horror of that, which I've already described, but knowing that there's nothing that can be done. And that is the hardest lesson of all. And we are spoiled by all of our advances in medicine, because for most everything that we can get, there's some at least initial therapy. Even if it's a terminal cancer diagnosis, there's ways that we can keep that disease at bay for at least a while. But for this, there was, at least in March, any knowledge of what could be done. And so there was a lot of flailing, and that concept of not knowing what's available, not having a treatment, was very sobering. But I anchored my thinking back on the patients who I took care of back in the 80s, who had the same experience. And I learned a lot from them, as we all do, always learning from our patients as we go. There's controversies, although I don't think it is. This is airborne, period. It's airborne. It is transmitted through droplets and droplet nuclei that are expressed from a patient who's infected. And one of the things that I think is critical to understand is that the peak time of transmission when somebody is the most infectious is in the 8 to 24-hour period prior to the development of symptoms. So in my case, when I was in the car with my son, in that 24-hour period, he was spewing lots of virus. And it would be almost impossible in a closed space with him not wearing a mask and me not wearing a mask for me not to have become infected. So there's no question this is transmitted that way. Despite whatever controversy is going on at CDC right now, this is transmitted predominantly, I won't say exclusively, but predominantly in a big way through the air. So what about masks? Well, here's a nice cartoon that shows this is a woman who's infected, and she's spewing virus into the environment around her, much like my son did in the car. And if this guy's wearing a mask, that helps a little bit, but you can imagine, unless it's an N95 mask, there's particles that are getting around the mask and into this airway. And this woman is 100% unprotected. So this is not a good scenario. When the source patient is wearing a mask, that cloud of virus is reduced substantially, probably 90+%. And so then that reduces proportionately the likelihood of transmission. And this guy now who's wearing a mask while she's wearing a mask is close, not 100% protected, but pretty darn well protected, especially if they're outdoors, not indoors, and they're not in very close proximity to one another where this can be transmitted. We have a good sense that a normal breathing, there's about probably about a 2-foot radius around us that where breathing will be putting the virus. But if we're talking, it could go up to 5 or 6 feet. If we're screaming, like if say we're at a football game or something like that, or in a bar talking loudly or laughing, that could go 6 to 8 feet. If we're in a choir room and we're singing, it could be 10 to 12 feet. And you can imagine indoors singing with no masks. That's where spread happens. And indeed, some horrible epidemics early on, in particular in Washington State, where out of 60 people at a choir practice, 51 became infected and about 3 died from that exposure. And it was just a 2-hour exposure. So masks work. If you want an image, those of you who are old enough to remember the Peanuts cartoon by Charles Schulz, this was a guy named Pigpen, and that cloud that's sitting around him in the case of COVID is the virus. You could think about moving it up a little bit to around his face, but you get the picture. So we can reduce transmission. So the summary, the common questions are, how is this transmitted? Answer, airborne. What can we do about it? We can wear a mask. And Dr. Redfield, when he testified at Congress about a week ago, he's there again today, but when he testified about a week ago, he said that masks are probably around as effective as a vaccine will likely be. And I agree with him. I agree completely. You put the two together and you've got a real great solution. But the vaccine alone may not be enough, especially if it's only 50% effective. But vaccine plus mask equals success. So think about influenza this upcoming season. If we're wearing a mask, we prevent COVID transmission, but we also prevent influenza transmission. And we also get a flu vaccine, and that's what COVID will probably look like if we're lucky, underscore that, and have a vaccine in the next 6 months, which is questionable in my opinion. But I'm hopeful, but I'm not ridiculously, I'm realistic. We've worked on AIDS vaccines now for 30 years. Every single one of the AIDS vaccines that went to a phase 3 study had great preliminary data. All of them were immunogenic. All of them had great antibody responses or cellular immune responses. All of them looked great. And then they put them into people and they didn't work in terms of transmission prevention. So we need to be pushing, but we shouldn't be crazy. We shouldn't count on a vaccine being, to use Greek tragedy terms, a deus ex machina, where it comes in at the last minute and saves us out of a horrible situation. We are in control of what we do right now, regardless of what happened to this point. Everyone should wear a frickin' mask, period. And if we did that, we'd have a shot. And if we blow it off and we act, in my opinion, stupid, and we don't do the right thing, we're getting what we deserve because we're not taking control. We're letting the virus rule us. And that's unfortunate. Hopefully we can change that. What about crowds? This is a really interesting analysis, and it looks at different case rates. At that time, when this was first done, Alabama was 39 per 100,000. Now we're probably about 20, 25 per 100,000. And you can discount this or go to the website on Georgia Tech. They have this website for estimating COVID. But let me walk you through this. So let's just take Alabama back then. If we have 10 people at an event, the probability that 1 out of the 10 will be infected with COVID is 40%. That 1 out of the 10 is 40%. If we go to an event of 25 people, the likelihood that 1 out of the 25 will be positive is about 70%. 50 people, 92%. 100 people, almost 99%. Get the picture? So for those of you who are thinking about weddings or going to large events, keep this number in mind. This is why we say avoid crowds, because the more people who are there, the higher likelihood that one person in the crowd, and you're not going to know who they are. Like I said, most transmission is happening before somebody gets symptoms, and we also have the people who don't have symptoms at all, like on a lot of college campuses and in K through 12, where transmission is happening silently and you don't know it unless you test. So avoid crowds. Avoid any crowd anywhere where people are not wearing a mask. And that's one of the reasons why around the world, one of the highest risk activities is going to a bar or a pub, because people crowded together indoors, relatively poor ventilation, and it's hard to drink a drink with your mask on. I guess you could put a straw in or something, but that sort of defeats the purpose, but you get the picture. All of this makes perfect sense. Well, let's move on to clinical presentation. For those people who get symptoms, the median incubation period is about 2 to 14 days, usually 2 to 5. In my case, it was 2 days. Most people, it's around 4 to 5, but that could also depend on the inoculum, right? If I got a pretty big dose of COVID, I'm sure, when I was in the car, and so I think it happened faster. As far as symptoms, these are overall symptoms here, but the key about fever is that it's like you heard in my case, it was present in the evening, but it wasn't present during the day. So if I had gone to an event and they tested my temperature going in the door, what good does that do? That's not helpful. If you ask me, have I had fever? That's much better. If you ask me if I had any of these other symptoms, much better, which is why the health check that a lot of us are doing at UAB, in fact, I think all of us are doing, is checking for symptoms, not measuring our temperature. There are a lot of unusual manifestations that I'll get into here in a second, but the course varies and every individual is an individual. About 80% of people with COVID who have symptoms do not get sick enough to go to the hospital. That was me, fortunately. But another 15% will go in the hospital but not get critically ill on a ventilator, and 5% will go on a ventilator. The case fatality rate is hard to know, to be honest. It's certainly higher than 1%, in my opinion, but it's probably less than 2%. But it just depends on how you do the numbers and how you incorporate asymptomatic people into the calculation. The point is, you don't want to get this because you don't know whether you're going to get sick or not. And if you're older, the chances of you getting sick, critically ill, and maybe even dying is much higher at age 70 than it would be at age 17. We know that. Does it mean that 17-year-olds don't die? No, 17-year-olds do die. I've seen it. We've all seen it. We've seen infants die. We've seen 2-year-olds die. It just isn't that common, but it happens. And so why even take the risk? Protect yourselves. Not only you're protecting yourself, When you prevent yourself from being infected and you go to visit grandma or you go to visit your parents, whatever it is, you're protecting them by keeping yourself uninfected. So what about prevention? Well, let's dig a little bit into the virus. We know that it comes into the system through the air. It binds in something called an ACE2, an angiotensin converting enzyme 2 receptor, and then enters into the respiratory epithelium. Once it's there, it's evil. It sets up shop like most viruses do and starts to produce its progeny, its offspring, and that's bad enough. But it also goes in the other direction and starts to burrow into the surrounding tissue, initiating inflammatory responses and initiating something that's really pretty horrible. In the pathology studies, the virus seems to jump into the bloodstream and then travel all throughout the body, the brain, heart, lungs, kidney, skin, you name it, it goes there. And then the immune system shuts it down, its replication pretty well, probably about day 5 or 6, but then the immune system actually is hijacked by parts of the virus in ways that we're not 100% sure yet. But that hijacking of the immune system is what I experienced. My immune system that shut down the virus, I'm pretty sure, but it was going out of control. The regulatory pathways that normally tell our immune system, all right, call off the guards, the fire is over, go home, aren't there, or they're not working right. And that sort of smoldering or that spiking cytokine storm or other problems are what are causing a lot of the morbidity and even mortality among patients. So we're developing treatments. I think we've put to rest finally the hydroxychloroquine story. There have been a lot of talk about, because of the cytokine storm, inhibitors of IL-6 and IL-1, such as anakinra. The tocilizumab, it doesn't, in clinical trials didn't work. Anakinra does appear to be working, but that's not 100% worked out. Maybe the tocilizumab was a problem because of when it was administered. And that's also true for convalescent plasma, another quasi-controversial thing. It's not that the science was saying it's a bad idea. The science was saying we didn't have it worked out well enough. What level of antibody is critical to have achieved in the donor for this to be worthwhile? That needs to be further worked out. And more importantly, and just like it is with tocilizumab, the timing, is it better to give it early, midway, late? Probably not late, but somewhere early, mid-level, that's when it's best used. We know that dexamethasone works, again, underscoring the fact that the immune system's gone awry. And dexamethasone is kind of like this giant blanket cooling off the immune system, not very specific and not very elegant. So that's where I think if we learn more about the immune response through our science, we can target therapies. I'm pretty excited, as is Randy Crone here on campus, who's like the guru internationally, actually, on cytokine storms. He thinks that IL-1 inhibition might be the way forward, and so forth. We do know that the virus, in addition to invading blood vessels, causes clotting, probably through an immunologic primary message. So almost everybody who comes in the hospital sick is getting anticoagulated to prevent any kind of clots, be it strokes or pulmonary emboli or deep venous thrombosis. Hydroxychloroquine basically, in well-controlled randomized studies, didn't work any better than placebo. And that's where the data are. There were a lot of studies that were published that showed in cohort studies that it worked better than controls, but the controls were not selected in a randomized way. It was people who they didn't feel like, either maybe they were too far along, whatever. But the point is that when you finally got the randomized data, it didn't work. Remdesivir did work, but not great. I mean, the data show better than placebo statistically. 5 days worked about as well as a therapy as 10 days, so that's what we do now. But we don't use it alone. We use it almost always with dexamethasone and some degree of anticoagulation. But let's look at the coolness of this study and of this drug. This drug was originally created back in 2015 in a partnership between UAB investigators, Rich Whitley, investigators at UNC, Ralph Barrick, and Gilead Pharmaceuticals, and I think some folks at Emory and Vanderbilt. And they were looking for therapeutics against Ebola, and it didn't work. And they said, well, maybe it works against the SARS, original SARS. And it seemed to have activity in some animal models against SARS-1, the one from 2003. So they kind of bookmarked that. When SARS-2 came along, SARS-CoV-2, this epidemic, they said, we've got a drug we think might work. So they did quickly some evaluations of this against SARS-CoV-2, saw activity, moved immediately into a phase 3 randomized trial. And by May, first part of May, they had this study done that clearly just demonstrated in a randomized controlled fashion that the drug had activity. But it's not a home run. It's a base hit, or maybe a bunt and you make it to first, to use a baseball analogy. But it's better than nothing, for sure. It's better used a little bit early than late, and it certainly works better when combined with dexamethasone. Antibody therapy I've already talked about. The case of dexamethasone, this was a randomized study, and the prevention of need for ventilation, the oxygen requirements, everything, this in particular was highly statistically significant and reduced mortality by about 10 to 20%, which is great. We've already talked a little bit about the immunopathogenesis. I want to go back to that because I think it's important. This is Sigma Chi, so you're really trying to study biology here. But as I mentioned, there can be therapies that might be addressed against the virus, blocking its entry through the ACE2 receptor. There may be issues about antigen presentation that we can interfere with or have some therapeutic and try to block the cytokine production or block the ability of the virus to shut off the immune system cool down messaging. Cellular immunity could well be important in any vaccine, and I think the AstraZeneca or the more live attenuated vaccines might have a better chance than the mRNA vaccines, which are the first time that mRNA has ever been used as a vaccine. And then humoral immunity against the spike protein of the virus in particular looks like it's going to be promising. Certainly the mRNA vaccines do a great job of eliciting lots of anti-spike neutralizing antibody, not just antibody, but neutralizing antibody, and that gives hope for the vaccine. That's like the Moderna vaccine, the Pfizer vaccines, which are now in phase 3. But again, can't help myself, got to caution that just because we see immune responses does not mean they are protective. And the only way we're going to know that is doing well-controlled, well-done randomized trials, which are underway right now. I'll skip this. I've already talked about it. I've already talked about the virus going into the bloodstream. These are little microclots here and microthrombi, and that's part of its pathogenesis and why it can be so difficult to take. I'm going to finish with this set of questions of what is called long COVID. And we're starting to see more and more of it, and it's pretty horrible. And as somebody who had this infection, I did not have much in the way of long COVID, but I did have some residual symptoms. What are they? I noticed that I can't walk up 3 flights of stairs like I used to without getting a little bit short of breath. I've had pulmonary function tests that are relatively normal, fortunately. I've lost my sense of smell, which is another symptom. During the time of infection, it came back, but not completely. So I can smell, but not like I used to. And it also, in my case, affected my hearing. And so that's been reduced some, and that hasn't come back too well. And that's very annoying, especially to people who want to talk to me. There aren't many of those, but some do. And it's annoying for me to have to say, I'm sorry, you have to speak up. So those are some residual symptoms. But what I'm talking about here are horrible symptoms, like not being able to walk more than 20 feet without having to sit down and rest 2 to 3 months after having the acute COVID illness. Chest pain, not cardiac in terms of blockages, but some sort of myocardial inflammation perhaps, or maybe non-cardiac chest pain. We don't know exactly how to characterize it yet. So these are things that everybody's exploring, but it's long-lasting effects. And the neurologic problems, I mentioned hearing and smell, which in essence are neurologic, but other people, this fuzzy thinking that I mentioned that I had during the acute illness, some people are having that and worse. A lot of them are having cognitive impairment. Maybe after you heard this lecture, you'll say, yeah, he had cognitive impairment too, but severe cognitive impairment. We could call it a form of dementia, but it's more selective, not quite as pan memory loss, but more selective memory loss and ability to function normally. So these things are persistent and are unknown as far as why. I suspect it's damage, probably immunologic that just hadn't got recovered. The question is, what can we do to prevent that in the first place? And once somebody gets that, what can you do? Fatigue is the number one. Do I have fatigue? Yeah, but I don't know if it's from having the COVID or just dealing with COVID. I think we're all worn out from dealing with COVID. I know I am. And the late nights get old after a while. But fatigue, shortness of breath, joint pains, chest pain, cough, I've mentioned a lot of this already. Some of the other concerns are the cardiac. We mentioned this. There can be an acute injury during the primary event that can lead to even heart failure or shock. There's the myocarditis that was talked about a lot, concern potentially for athletes. It's unknown how much that can happen. It's certainly not hugely prevalent, but it is a concern we need to evaluate. And then anytime you get myocarditis, you have the chance of arrhythmias, and I've already mentioned 3 or 4 times clots or thrombosis. So what do we do to prevent? Masks, keeping distance. You know the drill. I don't need to repeat myself. But one of the things we're doing on college campuses is a pretty innovative program where we call it Guide Safe. Those of you on our campus or in the state know about this. It's basically a comprehensive program that included return to campus testing. I'll show you those data in a second. It includes sentinel testing. I'll talk about that. It includes these health checks that we have on our cell phones that we want people to do every day and to notify if we've had an exposure. And then somebody who turns positive, or those who opt in, there's an ability to do contact tracing from the phone without identifying who the source patient was. We can simply find somebody whose phone was in contact within 6 feet of another person in the period of time before the person got sick for more than 15 minutes. This will notify that individual who was the contact, who came into contact with the index person, that they have had an exposure and should go get tested, without identifying who the person was who was recently diagnosed. As far as back to school, we did, in the matter of 3 weeks, 75,000 tests, very low prevalence, very low, but not zero. And it was pretty successful. But then the students came back to campus. This happens to be the University of Alabama. I'm not trying to pick on them. Other campuses, Auburn and some others, had similar things, but this is like 2 days or 3 days after return to campus. And I challenge you to find anyone in this crowd wearing a mask. And look how they're crowded together. And even though they're outdoors, this isn't something that I would recommend. And again, I can't count the number exactly, but it feels like around 50. And as I've already told you, the chance that at least one of these people is infected with COVID at the time they crowded together is around 92%. So there was a spike in cases at the University of Alabama. That's happened at Auburn as well. The good news is, through contact tracing, quarantine, isolation, it's now come down dramatically and I think is relatively safe there. We're also doing this thing called sentinel testing, which a lot of you will get notified about. People say, well, what is that? My best analogy is Hamlet. For those of you who remember this play, the opening scene was probably one of the best in all of literature, where a sentinel guard is watching out for enemies. And at the beginning of the play, he hears the rattling of the chains of the ghost of Hamlet's father, which is either probably off stage. And it has the best opening line of any play ever written. which for those of you who remember this, who's there? Who's there? And it's a brilliant opening line, not only for the play because it's responding to the ghost and say, who's there? But also if it's staged properly, we'll turn to the audience and say, who's there? As if, who's in the audience tonight? Who's watching? How am I going to interact with you? And so it's just brilliant, just a brilliant opening line. But the reason I like it as well is that this ghost is very much like the coronavirus. It's invisible, we don't see it, we know it's there, and it's a threat. And what we're doing through sentinel testing is we're trying to find out who's infected. One of my personal concerns right now, and I'm not taking shots, it's just the way it is what's happening right now in K through 12. Because it depends on the school system. There's a lot of desire to get back to normal. I understand that. It's critically important for students to have socialization. That's important, but we're also living in the middle of a pandemic. And the concern is that there's not any sentinel testing to speak of in general. And so we don't know what's happening within those schools right now. And my concern is that without the sentinel testing, it's like flying an airplane in dense fog or dense clouds with no instruments. We can't tell where we are. We can't tell we can't tell whether there's something in front of us that we're about to hit. We can't land the plane safely without the instruments. We need some degree of testing, in my opinion, in these environments to help guide us through this. Finish up with vaccines. At least 5 now are in clinical trials. A lot of discussion about Russia, who went forward with their vaccine, and it's now being released with no data. They're just, here you go, Russia. Citizens, take this vaccine, maybe it'll work. But the concern, of course, is its safety, not just its efficacy, but its safety. And I think that's just almost criminal to release any type of product, but especially a vaccine in the environment we live, where there are people who are afraid of vaccines. I know a lot of people who won't even take a flu vaccine because it may give them the flu. How in the world are we going to release a vaccine to the public without robust safety and efficacy data? We need that, and it needs to move forward undeterred. So I think I'll stop there. I will stop sharing my screen if I can figure out how to do that. And we will get back, and it looks like we have at least 15 minutes or so for questions. So I will, let's see if I can do that. How do I stop? Am I still sharing, or are we out of that?
You're still sharing.
Oh, hold on. Let's see. Shoot. It's not giving my... Go ahead and we can start answering questions while I mess around.
So, the first question I got was, what types of masks do not do a sufficient job of prevention?
Yeah, good question.
So which one do not?
Yeah, it's a really good question. And there have been some studies. The best mask is something called an N95, which if you're wearing it properly, you feel like you can't breathe. And that's what we use in the hospital for obvious reason, but it's not reasonable to use that on the outside. There's something called a KN95 mask, which is decent. But I think any mask, any kind of mask you might wear, get from a surgical type of situation. I think a well-designed cloth mask that covers nose and mouth will be fine. The one thing that's shown not to work too well is bandanas if they're thin. So that apparently doesn't trap enough to go through. Oh, here we go. Here, stop sharing.
We also have the second question, like, why are we not doing more sentinel testing? Policy, test shortage.
Messed up.
So that's my question.
No need to apologize.
Why are we not doing more
testing? The question is specifically sentinel.
Yeah.
Is it a question of a or a test shortage?
Yeah. So the reason, we are doing it on college campuses, so that's good. But it's about always money, and then it's logistics. So it takes a strategy right? It takes a plan. It takes a master way forward, somebody who's thought about this and issued it. Other countries, especially in the Asian Rim countries who suffered through SARS-1, or at least the threat of it directly, they were prepared. Canada, which had a few cases of SARS-1, took it seriously, and they prepared a plan. The United States had plans that were developed but weren't implemented, to be very honest, and at least not in a way that has been communicated. What our country has done so far is say, okay, here are some guidance. States, it's up to you to figure it out. Here's some money. They did send some CARE Act money to us, but it was up to us in terms of how to spend it and what to do. I think we're in the middle of a giant experiment, and because we've never faced anything like this in our lifetime. And right now, if I were the Grand Czar, I would say, okay, let's learn from the past 6 months, draw a line, and from this point forward, here's what we're going to do. 1, 2, 3, 4. And we're going to do this as a country together. And that's what it's going to take. So I'm going to try to avoid any kind of politics. I'm just going to state facts, in my opinion, about what needs to be done. And that's what I think needs to be done right now, regardless of it being an election year or whatever. That's what we should be doing. The longer we wait, if we think it's bad now, it's not going to be any better in January if we keep doing what we're doing.
Right. You know, I teach in clinical lab sciences, and, you know, and we have, if you have experience in the clinical lab, you can see that you don't need 10 different tests that have 500 kits or something. You want a lot of one, right? Because it takes a long time, like you said, to standardize, to, you know, to change your whole ordering and everything to test. So if everybody's creating a different test, that actually does not really help a lot, right? Because it makes the situation more complicated. Do we have like another question, any information from the Southern Hemisphere on co-infection of influenza and COVID?
Yeah, so that's a great question. Actually, the Southern Hemisphere did pretty well, especially Australia, New Zealand, where we often get our data for the flu vaccines and for other things. They had a very small influenza season, and it's because people wore masks. I mean, it's clear evidence that this works. So that's our lesson from that. We still aren't 100% sure of a co-infection at the same time between SARS-CoV-2 and influenza. I don't see any reason why it wouldn't happen. We have COVID infection with bacteria, with fungi, and so there's no reason why a virus wouldn't pile on in that situation. So let's avoid it. We know what to do.
I think we will wrap it up here. We still might have a couple of more questions, but I know that, you know, we have your time. You know, at this time, we want to thank everyone. And Maggie, are you ready?
Yes, thank you, Anna. And thank you, Dr. Saag, for this great seminar. I am Maggie Ansler, the treasurer of UAB Sigma Xi. And for those of you who aren't familiar with Sigma Xi, it's a pretty old organization founded in 1886. And the founders emphasized that this new honor society would be broad in its outlook, devoted to all science and engineering, and designed to reward excellence in scientific research, and finally to encourage a sense of companionship and cooperation among scientists in all fields. So in selecting the name Sigma Xi, in part because these Greek letters weren't being used by any other organization at the time, but eventually a motto was established based on the letters, and it confirms the purpose, which is Spodon Xynones, or companions in zealous research. So Dr. Saag, your career of research dedicated to studying viruses afflicting the global human condition sort of exemplifies a deep passion and zeal in understanding the genetics and transmission, mitigation especially prevention of viral diseases, especially HIV and now coronavirus 19. So in recognition of your efforts, both literally and colloquially viral, Sigma Xi UAB is proud to present you with an honorary membership in the society. On behalf of the chapter, thank you for your zealous research efforts in the fight against deadly viruses. And I'd like everyone to join me in a round of virtual applause for Dr. Saag. Thank you.
Yeah, maybe we can go ahead and put our little hands. I'm not that that versed. I think for some reason he dropped, or maybe, he's not here. But we'll make sure that we send him this email. Something happened. But we want to thank everyone. And, if you want to be in, you want to participate in our chapter, if you want to know more about it, just go ahead and email us. We'll have another seminar on October 21st. So thank you, everyone, and stay safe. Bye.
Thank you, Rachel.
Yes, thank you, ladies. I hope you both, all three of you, sorry, have a wonderful day. All right, and I will get you. Okay.
You're stopping.
Thank you.
Yeah.
You're
All right.